For this video, we will cover the methods of microbial count. There are a couple reasons why a person may want to perform a microbial count. In laboratory settings, this could be for wanting to know if a sample has grown, or maybe wanting to map out growth patterns. Another big reason we care about microbial counts is for consumer purposes. Here, food or beverages that are to be sold need to have a low enough level of microbes or types of microbes to be safe for consumption. And we can use microbial counts to ensure that we reach these levels. Alright, let's go ahead and cover some of our techniques. The first technique is the plate count, which can be done in two ways. So let's just go ahead and cover those real quick. For both types, a serial dilution is needed where the sample is diluted multiple times in order to limit the number of microbes in each subsequent tube. Then we can either do a pour plate method or a spread plate method. Let's start out with the pour plate. In the pour plate, a small amount of the sample, about 1 ml from each tube, is placed in a blank petri dish and sterile agar is added to make up agar plates. After incubation, the plates are examined for growth. Note that we only count plates that have between 25 to 30 and 250 to 300 colonies. The numbers depend a bit on who you ask. We can then use the numbers of colonies to back calculate original cell density, or OCD, or the number of microbes in the initial sample. The basic equation for this is that the OCD is equal to the number of colonies divided by the sample volume multiplied by your dilution factor. So as an example, let's say that we use our third tube, which would have a dilution factor of 1000. The sample size was 1 ml and the number of colonies is 32. The equation would then be OCD is equal to 32 divided by 1 multiplied by 1000. So our OCD is 32,000. Depending on the sample, this can then be compared to FDA guidelines to see if it is safe for consumption. The spread plate is similar, except here we would already have plates made, and we then add the 1 ml of sample to the top of the plate. We then spread the sample on the plate, and the sample can be counted after incubation. Another technique is filtration. Here, a sample is passed through a filter that collects the bacteria. The filter is then transferred to a petri dish and grows colonies on the surface. So, as an example, let's say we filter 100 ml of milk through a filter and place it on an agar plate. There are 150 colonies after incubation. We would then say we have 150 bacteria per 100 ml of milk. A third technique is what we call the most probable number count. Here, multiple serial dilutions are completed, and after incubation, the number of positive tubes within each set of the serial dilutions are then counted and compared to a standardized chart for the approximate volumes and sample sizes. In our example here, the score is 521, and if we were to look at the table, we would have a MPN index of 70 with a lower limit of 22 and an upper limit of 170. This means that with a 95% statistical accuracy, we can say that our sample will have between 22 and 170 bacteria per 100 ml, with 70 being the most probable number. A fourth method is the direct count. Here, a sample is added to a special microscope slide that has a grid on it. The bacteria are then directly counted, and you can get the number of cells per ml simply by dividing the number of cells counted by the sample volume. This method can be good for quick counts as the results are instantaneous. The last method is the turbidiometric method. Here, a spectrophotometer is used to calculate the absorbance of light, which can indicate different levels of growth. You would start by adding a blank sample of media to calibrate the spectrophotometer. A sample is then shaken and placed in the spectrophotometer, and if it does not have a lot of growth, a lot of light will pass through and absorbance will be low. On the other hand, if there is a lot of growth, then little light passes through the sample and absorbance will be high. This concludes our video covering methods of microbial count.